This recorded session is the last part for our module on B and T lymphocytes. So I've already mentioned about how antigen binding activates lymphocytes, especially for the case of T cells. For example, you would remember how effector helper T cells differentiate after they are presented with antigens in the presence of the appropriate cytokine, of course. Diba? I think I've mentioned about that in our introductory recorded session. No? The different effector T cells that mature based on the present cytokine in their microenvironment and the nature of antigen presentation that happened. No? Now let us examine more closely the interaction between T cells and antigens. Immature or naive T cells, as we call them, immature naive CD4 T cells, they will commit to being T helper precursor cells, and once that they mature, no, into to activated ones, they are ready to secrete a set of cytokines that are important for other immune processes. For this maturation to occur, no, antigen presentation is a must. So antigen presentation is necessary for the maturation cycle or the maturation processes to actually be activated. Helper T cell activation starts with the interaction of the cell with an antigen presenting one, with an antigen presenting component. And in terms of location, in non lymphoid tissues, the APCs include phagocytic and dendritic cells, while in secondary lymphoid tissues, the resident dendritic cells carry out antigen processing. APCs present the antigen MHC complexes bound on their surfaces to the CD4 T cells in order for activation to occur. In addition, with with antigen presentation, we also have co-stimulatory molecules that participate in T cell activation. Co-stimulatory proteins such as for the example I have here on this slide, CD28, no, they are expressed by T cells and they contribute to the generation of important intracellular signals to increase binding affinity. The receptors of these co-stimulatory proteins, they're found on the antigen presenting cell, on the of course the interacting cell. So you expect that as the APC presents the antigen, the T cell co-stimulatory protein will also look for its receptor on the APC. For the case of CD28, it appears to actually regulate a signal transduction pathway that is particular to T cells, particular to distinct to TCR activation, and it has a major role in the induction of cytokine production and the expression of certain survival genes that are important for the stimulated cell to contain to important for for the longevity of the stimulated T cell, no, for the case of CD28. Now, in the absence of co-stimulatory proteins, even though APC presented the peptide MHC complexes to our T cells, no, in the absence of these co-stimulatory proteins, T cells may still be dormant. They will not really activate. They may become refractory to stimulation, meaning even if there's enough stimuli in their microenvironment, they will just behave in a manner that they won't continue maturing. They will not really proceed with effector differentiation. and then in some cases they will die. No? Now, since we've mentioned, or since we've already uh, closed the chapter on T cells, no, let's take a look finally at the molecular events underpinning signal transduction in T cells. Kasi essentially this activation, no, uh, we describe it in terms of the elements, the cellular and molecular elements that play major roles for the cell to be for T cells to be fully activated. Now, will we have to also understand in terms of the molecular terms, or in terms of the molecular players, how the activation process will actually, how the activation processes will actually happen. No? So what effectors are involved? Okay. Um, okay. So, but actually, before we proceed to that, no, a quick mention about co-stimulatory proteins. So, for the case of T cells here, CD28, no, CD28 is a very complex co-stimulatory protein. The receptor for it may actually be a variety may come in more than one set, no, or more than one individual receptor. Like for the case of CD80, CD86, no? CD28 can actually bind to such receptors. Okay, and what else? Yeah, actually, it's everything I just have. I really need to mention about co-stimulatory proteins. Anyway, okay, so now let's move on to our discussion about signal transduction in T cells. Okay. Um, um, okay, so for the longest time, okay, we know how, or for um, actually in the first parts, no, of our discussion for the second quarter of our entire course, no, I've mentioned about tyrosine as an important amino acid. And now we're going to appreciate how important tyrosine is, especially enzymes that are involved in tyrosine chemical reactions. Tyrosine kinases. So in this whole pathway, we can see the production of these important enzymes. Actually, this figure here, it illustrates how inositol lipid components, inositol lipid compounds, are metabolized inside T cells. The combined interaction between antigenic receptors that are bound to the TCRs and the co-stimulatory proteins such as CD28 triggers cytosolic tyrosine kinases. So kinases that are within the cell. And these kinases, they are linked via adapters to certain effector enzymes, such as phospholipase C, and we also have phosphoinositide 3 kinase. No? So before we proceed to the remainder of this illustration, no? So just a review, a tyrosine kinase is an enzyme that is capable of transferring a phosphate group from ATP to tyrosine residues of a specific protein inside the cell. So you remember phosphorylation as an important event in the activation and deactivation of molecules. So for the case of signal transduction, no, major enzyme doing activation and deactivation are actually tyrosine kinases. These two enzymes, phospholipase C and um, um, phosphoinositide 3 kinase, they act on a single substrate, which is phosphatidyl inositol bisphosphate. This one. Okay. Phospholipase C will hydrolyze this substrate to produce IP3, inositol triphosphate, and diacylglycerol. On the other hand, our PI3K will process this substrate in order to produce phosphatidyl inositol tris phosphate. So, um, addition of a phosphate group. These mediators here, okay, they or these what do you call this? These precursor molecules. Okay, they will give rise to certain kinases, tyrosine kinases that do their thing, no? They're activating or deactivating cell molecules. Okay, so inositol polyphosphates, in the case of IP3, and actually all of them naman, IP3, diacylglycerol, and phosphatidyl inositol 3-phosphate. All of them are signaling molecules, no, that transduce signals from the membrane of the T cell, where TCR antigen interaction occurred initially, and then to the interior of the cell. Most definitely targeting yung gene expression, targeting necessary up or down regulation of certain gene byproducts. Okay, so these kinases, they have also, in addition, I have to mention this, these kinases that are produced ultimately from the breakdown product of other um, uh, 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 polyphosphates. Okay, they function in localized parts of the cell. Some can function within the cytoplasm and the nucleus. Others can function on the membrane and the cytoplasm. Okay, like for the case of protein kinase D. Probably you have encountered protein kinase B and protein kinase D, no, in your biochemistry classes. Protein kinase B is actually a serine-specific protein kinase, and it plays multiple functions, such as in glucose metabolism, apoptosis, cell division, transcription, and movement of cells, no, from one area to another. Protein kinase D, on the other hand, it's actually also a multiple, I don't know, a multifunctional kinase. It's involved in cell division, cell commitment, differentiation, membrane transfer, or no, membrane enabled transfer of chemical compounds, secretory functions of the cell, immune regulation, definitely, and in some cases, I don't know, mga pathologies like cancer. Okay, now um, we've finally closed the chapter on T cells. So to of course balance things, as we've mentioned, or we've been discussing T cells and B cells for almost 3 recorded sessions now, no? Let's also take a look at how B cell development actually progresses. B cell development takes place in the bone marrow and the spleen, and it can be subdivided into different stages based on the expression of different cell surface molecules and intracellular markers, as well as, of course, very important, no, the rearrangement of the cell's heavy and light chains, no? The rearrangement of the genes coding for the heavy and the light chain components. No? In terms of duration, the whole development process can take 1 to 2 weeks, starting from hematopoietic stem cells until eventually you have an acceptable population of immature, naive B cells. No? Similar with T cell development and maturation, of course, the goal of B cell development is to create a repertoire of receptors, diverse receptors, capable of recognizing a plethora of antigens, while at the same time eliminating those that are self reactive, those that are autoantigenic, no? Those that respond to autoantigens. So in a snapshot, B cell development follows several stages. Let's take a look at this um, wonderful illustration. So B cells, they arise from pluripotent stem cells, similar with T cells, that are initially when we are still developing, no, the progenitors of the stem cells, the hematopoietic ones, they are initially produced in the fetal liver. Then eventually, as we mature, it's the bone marrow that takes place, no, or that takes over the production function. Okay, so B cells arise from these pluripotent stem cells. Then the stem cells progressively develop into pro B cells, pre B cells, immature B cells, and transitional ones, no, which are essentially which are located on the periphery, or has peripheral spatial distribution. Okay, um, the final product, of course, is a mature B cell that expresses a functional BCR, a functional B cell receptor. These different stages of B cell development, if you are to study them, you can differentiate them according to the status of the immunoglobulin genes expressed by the developing cells. So, pro B cells, the IG genes are unrearranged. So ano, yung original na conformation, no? Germline yung status ng mga immunoglobulin genes in a pro B cell, no? Um, Now, in the pre-B, the pre-B cell stage, we expect the rearrangement to already start. Okay, so DJ, VDJ rearrangement are expected to happen at the pre-B cell stage. And aside from IG gene rearrangements, pre-B cells also are characterized with the expression of cytoplasmic IG heavy chains, no? IG mu heavy chains. Okay. Then immature B cells, they express IgM on their surface as an indication that from pre-B cell you already have an immature B cell. And lastly, transitional as well as mature B cells, they express both IgM and IgD on their surface. In a while, we will discuss them with additional details. The sequential expression of cell surface receptors and adhesion molecules that demarcate or differentiate the different stages of B cell development allows for the B cells to actually undergo certain functions, no? Such as for them to proliferate or to move from their original location, from the bone marrow, no, to locations such as the spleen, where they can experience further, I don't know, maturation through antigen presentation or um, antigen interaction. Okay. The loci for the heavy and the light chains are also arranged in a particular order. It's not only, the cell surface receptors and molecules that appear in sequence. We also have how the IG genes being rearranged in a particular order. The heavy and light chain immunoglobulin genes, they are arranged during the pro and the pre-B cell stages, respectively. And um, to top the rearrangement, we have surface IgM expression as an indicating or as a signal event. Okay, so surface IgM tops that, or signals na tapos na siya, no? Further developmental progression and maturation in the periphery, in the peripheral areas, are limited by positive and negative selection, which is mediated by BCR signaling to produce yun nga, mga functional B cells, no? with BCRs that are non-self-reactive, those that we can consider as immature, naive B cells that are ready to mature in a little to they're ready to mature once that they encounter foreign pathogens or antigens. Okay. Now, to help us remember some of the finer details of B cell development, I have here a very nice illustration as well, no, that I want you to review. So you can associate your own spatial distribution with the surface molecule expression, surface protein expressions, as well as Ig gene rearrangements. No? So early B cell development constitutes the steps that lead to B cell commitment and expression of surface immunoglobulin. So essentially, or in a nutshell, we expect that the mature B cells they will leave the bone marrow and they will migrate to secondary lymphoid tissues, so that, or not really mature, but more of the B cells that have accomplished the necessary Ig gene rearrangements, as well as surface BCR expression. They will leave the bone marrow. They will migrate to peripheral areas, to secondary lymphoid tissues, where they expect to be where they are expected to interact with foreign pathogens, with antigens. No? Okay. In during such interactions, no, B cells may interact with exogenous antigens. Okay, and such, they can be activated on their own, no, pwedeng without the help of any other cell or any other immune effector. Also, some of them can or will require the assistance of T helper cells for activation. No? Okay, then as of course in the peripheral areas, this immature naive B cells, as they enter the last part of their differentiation, whether they will become a memory cell or a plasma cell, no, we expect a negative selection to occur. So BCRs that are of no potential, or B cells with BCRs that are of no potential value, should be deleted, or if not, if not, the receptor can be edited so that it can be useful. No, the B cell can become a transitional or mature B cell that is possessing a BCR that can still work at particular uh, episodes, that certain antigens that or antigens that are yet to be met, no, are yet to be processed by the B cell, or yet to be recognized. Okay. Okay, so in summary, here are the developmental stages that B cells undergo, starting with hematopoietic stem cells. Okay, so we have to be able to differentiate now where T cell and B cell production will actually split. No, so we have here a sequential list of the different stages. On ano to, no? Of course, we're done with the molecular part, with the specific details per cell. So let's review them. We have hematopoietic stem cells, which are the self-renewing, um, pluripotential. No? They give rise to all blood cells, as we know, and depending on the target, the stimuli that it receives, different transcription factors can drive maturation, can drive commitment of hematopoietic stem cells. Then we have multipotential progenitor cells that lost the capacity to self-renew, but they can still differentiate into different lineages. No? Hindi pa masyadong restricted. Then we have our Lymphocyte primed multi-lineage progenitors. So they express certain molecules such as FLT3, which bind to interleukin 7 receptor. Which binds to its ligand, FLT3 ligand, and once that an LPMP meets or is bound to an FLT3 ligand, it will proceed to IL7 receptor synthesis. Okay, the FLT3 expression will actually marks the loss of multipotentiality in LPMPs. No? ELPs, the fourth stage for B cell development. So these early lymphoid progenitors, a common molecular marker among them, kasi early lymphoid progenitors are actually the diverging point between T cell development and B cells. No? So for ELPs, the molecular marker that you should be remembering are recombination activating genes, RAGs. So recombination activating genes, which are useful during the gene rearrangements. And then these early lymphoid progenitors, some of them can migrate to the thymus, giving rise to the T cells, while others stay in the bone marrow, giving rise to our B cells. Now we have the common lymphoid progenitor, which um, can mature into natural killer cells, dendritic cells, B cells. No? These common lymphoid progenitors are responsive to IL-7, okay? And they have already lost their myeloid potential. Okay? And then the common lymphoid progenitor will give rise to, for B cell development, we have our initial stages, our pro-pre-B cells. Okay? Now, during the early stages of B cell development, similar with T cell development, functional rearrangements of the heavy chain gene locus allows for the assembly of the pre-B cell receptor. No? And normally, after that is accomplished, there will be a stoppage on the rearrangement. So there will be a signal to stop further rearrangements of the Ig gene for the heavy chain. No? So that process we call allelic exclusion. Then afterwards, light chain rearrangements will begin. No? So light chain rearrangements will occur, and eventually to top the gene rearrangements, the production what do you call this? Um, um, your the components of your the components of your what do you call this, of your BCR is your BCR, which include what do you call these, yung other chains ng ating surface IG, they will participate, or they will be involved in the initial arrangement of the receptor complex, so that um, once those chains are already have started to be produced, mag-stop na yung rearrangements for the light chain. Okay. In pro B cells, the Ig gene rearrangements, they are initiated by a juxtapositioning of the J segment in the heavy chain, okay, to a D segment. So magdidikit sila, no? There is a combination of a J segment and a D segment in pro B cells. Okay. Subsequently, all right, a V will be attached. A V segment, variable segment, will be attached to that initial DJ rearrangement. So that will give rise to that VDJ. Okay, VDJ rearrange gene segment. Okay? Now, nasa ano na siya, no? Nasa pre-B cell, pre-B cell stage na siya. So this will result to the generation of diverse receptor, diverse, what do you call this, variable component, no, nung ating uh, membrane bound immunoglobulin, or our BCR. Okay. And after the arrangement process, the IG gene, the IG gene rearrangement for heavy chain has been completed. Of course, the same thing is expected to happen for the light chain. Diba? Successful rearrangement of both IG gene for the heavy chain and the IG gene for the light chain will now contribute to the surface expression of the intact immunoglobulin that will, of course, be mounted on the surface. Okay, our naive B cells, with the expression of the intact surface IgB cell receptor, will now enter a state at which it's ready to seed, let's say it's ready to undergo further no, maturation in the germinal center of lymphoid tissues, of secondary lymphoid tissues, where we expect antigens from the environment, antigens from the surrounding, are expected to be presented, no, to our naive and memory B cells. When the B cell receptor engages an antigen with the right specificity, the encounter will actually induce what we call an SHM, or a somatic hypermutation. So an SHM is an event confined on the germinal center, okay, of our secondary lymphoid tissues. It gives additional, or it adds additional diversity dun sa ating mga BCRs, no, or dun sa maturing population of our naive B cells. Okay, those mature naive B cell na napresentan na ng antigen, no, nakakilala na sila ng antigen, but they did not choose to go to the germinal center for them to further specialize, no. They will direct themselves to peripheral circulation, and then short-live yung activity nila most of the time, and they don't undergo, or they don't experience the wonders of somatic hypermutation. Okay. Okay. So now, so much about IG gene rearrangement. This is actually a very complicated narrative. No, I really just wanted to introduce the basic concepts, so that in your advanced immunology, this is something that you don't have to look back, or you don't have to review anymore. Okay. So for mature B cells, we can describe mature B cells as having complete IgM molecule on mounted on their cell surface. So the complete IgM molecule on their cell surface already marks the terminal stage na dapat nandun na ang mga mature B cells. Some resting populations of mature B cells, they express a variety of surface molecules like the ones here on the first sub-bullet. And they lost eto mga to naman, cell surface molecule na to that they don't express anymore. So you see, RAG1, RAG2, the recombination activation genes, they should no longer be expressed by mature B cells. Okay. So the mature B cells, their subsequent act is to exit the bone marrow, to exit what do you call this, the secondary lymphoid tissues, and then they will be ready to do immunosurveillance or antibody secretion. They can recirculate between blood and the lymphoid organs. They can enter follicles in our lymph nodes and in our spleen. They can respond to any antigen encounter, that could be T cell dependent or not. And they can definitely, of course, most important function ng mga B cells natin is antibody secretion, antibody production. Okay? So speaking of antibody production, we have our plasma cells, which are one of the potential fates of B cell differentiation. Plasma cells secrete antibodies and they have a few surface antibodies themselves. Those that arise from the follicular B cells or from the follicular region, they're found mainly in the bone marrow and they are long-lived. So, mas matagal yung kanilang residence time. However, those that arise from non-follicular cells, those that did not really undergo germinal center maturation, yung mga did not really choose to specialize further. Normally, they are short-lived, days to weeks ang kanilang residence time. And similar with, of course, our mature B cells, plasma cells, will also have specific cell surface markers on them. They will lose cell surface markers like these one, like this set. And then ito namang mga cell surface markers na to, they will definitely have this. Okay? Now, we expect that after an initial encounter with a foreign pathogen or an antigen, secondary memory cells will be kept in the circulation. And these memory cells, compared to the naive counterparts, have different characters. In terms of lag period after antigen exposure, memory B cells are faster than the naive ones, 1 to 3 days na lang compared to almost a week before naive B cells can actually be activated after antigen recognition. Also, memory B cells have a peak response time of 3 to 5 days. So mabilis din silang maging hyper active. It will only take a few days before they reach their peak activity. Then in terms of magnitude of the peak antibody response, compared to naive B cells, memory B cells are 10 to 1,000 times faster. Now, in terms of antibody isotype, the naive B cells we expect IgM, while for memory, those that are for secretion, normally IgA in mucosal tissues, and of course the most numerous Ig, we have IgG. In terms of lifespan, we have memory B cells to live longer compared to naive B cells, and both recirculate. Antibody affinity memory B cells are very interacting, very dynamically interacting with antibodies. Okay. All right. So in terms of activation, naman, we have several mechanisms, and the one that I think I've mentioned already was T cell dependent B cell activation. I think I've made a quick mention about this in some of the recorded sessions before. T cell dependent B cell activation is where your antigen specific T cells and B cells interact with one another. Normally, nasa in secondary lymphoid organs or tissues, where under the influence of additional signals provided by the lymphoid cells, by the lymphoid microenvironment in the form of the other cells, yung mga antigen presenting, mga stromal follicular cells, B cells become activated and they can enter either the extrafollicular reaction, which I mentioned a while ago, or they can also undergo further specialization. They can go to the germinal center. The extrafollicular reaction, it will yield our short-lived plasma cells that can secrete predominantly low affinity IgM. While those that finish germinal center residence, they will generate long-lived cells. They will be responsible for long-term immunity, long-term humoral immunity, and they essentially they are high affinity memory B cells, and memory B cells as well as the antibody secreting counterparts. On exposure on the same to the same, after an exposure to the same immunizing antigen, memory B cells, they can rapidly differentiate to become, the plasma cells. Memory B cells and plasma cells, they're derived from a germinal center reaction, where, yun nga, na-introduce ko na yung idea ng somatic hypermutation, which will result to an increased affinity for antigens and immunoglobulins. Can actually be the production, yung production, yung nature of immunoglobulins that are produced, they can be switched across varying types by cells that underwent GC reaction, germinal center reaction. Okay? Now, we've accomplished discussing B cell development and activation. So that's another chapter close. No? Now, to top our discussion, let us include some key points on the immunology of COVID-19, especially that there's some interesting work on immune profiling of patients that were positive for COVID-19, no? And patients that are currently that were positive for COVID-19. There were papers published about the immunology of COVID-19. No? So if you would remember, if not, no, I would be providing a general description on the immunopathogenesis of SARS-CoV-2 virus. No? So the S protein that is present in the membrane of our SARS-CoV-2 virions is the one considered as the most potent entry determinant of the virus inside the host cells that express the ACE2 receptor. Diba? So that S protein is actually yung antigen niya, no? Or not, not antigen, but the ligand, okay, that serves as as a key, okay, to host cells, allowing the entry of SARS-CoV-2 virions. There are a lot of cells in our body that express ACE2. It's a general receptor kasi. So in particular, in our lungs, as well as in our intestines, no? The cells here express ACE2 receptors. So you expect the SARS-CoV-2 virion to actually be capable of entering cells of the lungs, of the respiratory system, as well as the intestinal, no, gastrointestinal system. After the virus releases its viral RNA genome in the cytoplasm of the cell, in the cytoplasm of the host cell, structural proteins and polyproteins are produced so the virus can actually replicate. Upon infection with the virus, the host cell activates a well-coordinated and rapid immune response. Of course, we expect that. The innate and adaptive immune responses. In the endosomes, where your virus can probably be still encapsulated, endosomes within host cells or phagocytic cells, membrane-specific pattern recognition receptors can recognize the viral RNA as a pathogen associated molecular pattern. These are concepts I've discussed in our introduction, diba? We have for the case of direct antigen recognition, we have PRRs, pattern recognition receptors that are built in the genome of the vertebrate animal, of the mammalian model system. These PRRs can recognize microbial antigens as an indication of pathogen breach, no, of pathogen entry. Okay, interactions between the host cell and the pathogen, the virus, will activate either cellular immunity, where there's direct killing of virally infected host cells or There's also secretion of stimulatory molecules such as cytokines, no? And then we also have in cellular immunity, T cells that are T cell effector T cell maturation specific to specific to SARS-CoV-2. Then for humoral immunity, of course, we expect antibody production against it. That could be a B cell that could be B cell activation enabled by T cell T cell presentation or T cell what do you call this T cell supplementation. No? Now, yun nga, if a B cell is activated by a specific antigen or by the SARS-CoV-2 antigen to be particular for this discussion point. Now, we expect plasma cells, memory B cells specific to the virus, specific to SARS-CoV-2, to be produced. No? Um, the immunopathogenesis of SARS-CoV-2, one of the components that have been very much well discussed is cytokine storm. So a cytokine storm is when different cytokines, most of the time, inflammatory cytokines, different inflammatory cytokines are produced at a much higher rate than compared to what is normal. This overproduction will result to a positive feedback on other immune cells, and it will allow other immune cells to be recruited on the site of the entry of the pathogen. And instead of actually fighting the pathogen, it will cause additional tissue damage. And eventually might lead to not only localized tissue corruption, but also the entire organ will be damaged. Okay, so that's how SARS-CoV-2 actually tricks our body, and how it can cause one pathway can actually cause a detrimental consequence once that you're infected by the pathogen. Now, generally, for viral infections, Th1 cells mediate the immune response. They're very predominant in the adaptive immunity against viral infections. T cell responses, they depend upon the presence, of course, of antigen-presenting cells mediated by the additional chemokines in the environment. CTLs are capable of secreting a cluster of molecules that can be used for direct killing of virally infected host cells like granzymes, interferons, and perforins. Okay. Now, CD4 are helper T cells. They facilitate the overall adaptive response, no, by assisting cytotoxic T cells. So we already know the relationship between CTLs and helper T cells, diba? Then we have for B cells naman, they play a protective role by, of course, being the producer of neutralizing antibody. No? And they can also impede reinfection, especially if we have a very good titer of memory cells or competent population of memory B cells that can be activated anytime once that there's secondary or tertiary or succeeding exposures, to um, the same pathogen. Now, in one paper that I've read from Frontiers, in COVID-19 patients, there is an elevated level of chemokines and plasma cytokines, which actually corresponds to what we call nga as the cytokine storm. And if this is not managed in a timely manner, this can really result to lethal consequences for the infected patient. Okay? So moreover, like in cancer and other chronic infections, the persistence of cytokine storm will also not only damage the tissues but can also decrease the number of the immune cells. Like for the case of T cells, T cells may experience exhaustion and decrease in their population because of the cytokine storm. And eventually, everything boils down to to viral sepsis. Okay. um, and other failures will proceed. Okay, and death might be the final or the terminal consequence. Okay. Now, post-infection, what happens? And now that we've somehow uh, recognized, no, the how immune cells, how the lymphocytes participate in COVID-19 infection, how they behave rather, if a patient, if a human is infected with COVID-19 post-infection, what happens? No? There's a nice paper that was published around the same time this last year, that where they profiled COVID-19 patients in terms of the effector cells and lymphocytes. No? Um, Patients were divided into, or patients of various categories, patients that are early recovering and late recovering. They were compared against healthy controls, and then the immune cell profile of a COVID-19 recovering patient was established, no, by this paper. The authors report, no, that in COVID-19, after or post COVID-19, or during COVID-19, no, we expect nga yung exhaustion of lymphocytes, especially T cells. So um, during the early recovery stage, we might not see much of the T cells, according to them, but there's increased in monocytes and progenitors of T cells. Also, there was an increased ratio of classical CD14 plus monocytes with high inflammatory gene expression, as well as a greater abundance of CD14 IL1 beta monocytes in early recovering patients. No? So the T cells are not really, or the T lymphocytes of ERS, of early recovering, what they call early recovering patients, if they were compared to the control, significantly mas mababa sila. Also, CD4 T cells and CD8 T cells, our helper and cytotoxic, they decrease significantly, and they express high levels of cell, no, inflammatory genes. So you can still see yung mga traces of the of the inflammatory activity that COVID-19 is actually well associated with. No? That even in early recovering patients, still the genes for the production of inflammatory chemokines still active. No? Among the B cells, the plasma cells increased remarkably, whereas the naive B cells decrease. No? So here we can see a side-to-side comparison of the patients or samples that are of healthy control, mga early recovering patients and the late recovering patients. So you can see a cycle na, of course, during the early recovery stage, decrease in the lymphocytes is very evident. T cells, both helper and cytotoxic, and then yung mga naive B cells also are decreased, but B cells but plasma cells, antibody secreting, of course they are increased in significant proportion. Okay, so here we can see the immune profile of an early recovering to a late recovering patient. No? So you can expect that this should follow a cycle. Now, eventually, your fully recovered patient will have this immune profile again. So now, imagine, no, kung we have what do you call this? We introduce or we become vaccinated. No? So we mimic the entire process of immune response facilitated by T cells and B cells, and then without the fatal consequences na needs clinical management. No? So A lot of papers already are, or a lot of not, I'm not sure with publications, but a lot of people are already reporting, even medical doctors themselves, reporting yung ano, no, result ng increase ng antibodies nila after vaccine inoculation, after being vaccinated, even yung first dose pa lang. Okay? So that ends our discussion for this module. So I hope that we are able to, appreciate the very fine details of B and T cell development, as well as their activation and immunology.